And we're going to talk about our final Pillar 2 program for today, which is our CRISPR gene editing platform that is not viral delivered. So this is the first non-viral delivered CRISPR gene editing platform for the brain that we're going to talk about. This is that NIH grant that I told you earlier that has been one of the most remarkable and, exciting times of my new drug development career is working with these two and their whole team who are here. This has been one of the most exciting programs I've ever seen, something that's never been done before, and at time and time again, it just works, and it's pretty incredible. So Dr. Yong-hui Jiang and Dr. Jiangbing Zhou are here from Yale University. They're going to talk to you about this program. We talked about it a little bit last year, and they're just going to go over the progress that we've made, working closer and closer to get toward clinical trials.
First, thanks, Alison, for the introduction. And good afternoon, everyone. My name is Jiangbing Zhou. Slides on. Okay. So in the next 20 minutes, Yong-hui and I are going to talk about our work relevant to non-viral delivery of genome editing therapy for Angelman syndrome. So this is our disclosure. Both Yong-hui and I are co-founders and shareholders of Courageous and affiliated with Courageous. So we have been working on this project for many years. So today we have 20 minutes. So we mainly focus on the translational efforts, which is being supported by the NIH grants, as I mentioned a while ago. So for this grant, for the preclinical phase, mainly will be led by, it's being led by Yong-hui, and the clinical phase we are led by these Barry Kravis, so sorry about that. So I'm sure probably most people here know her very well. So in addition to the PIs, we have large investigators and other support staff at Yale working very closely with us for the translational efforts at this moment. And of course, very importantly, we work very closely with the FAST Foundation, mainly Alison and Jennifer, and also Courageous CSO Conrad. For all, this is really a large project, a lot of efforts. To be honest, Yong-hui and I could not handle this project without all the support from the FAST and Courageous. So I haven't told you my role on this project yet. So actually, when I stood on the stage last year, I told you my role is an Uber driver. Actually, my role is a little bit beyond Uber driver. I'm an engineer on the team. So I'm the person who creates the vehicle, which is designed for delivery of the genomic payloads to the disease target for Angelman syndrome, mainly neuronal cells in the brain. So therefore, in the next few minutes, I will talk about why and how we deliver this delivery system, and what are we going to do next. So first, a very brief overview of gene therapy. I know all the approach has been mentioned by previous speakers already. So there's three different types of gene therapy. The first one called gene replacement therapy. So in this case, one can deliver genetic payloads, either like DNA or RNA, into disease cells using viral vectors or other non-viral vectors. Relevant to Angelman syndrome, we can potentially use AAV or lentivirus, right, to deliver UBE3A to disease cells. So the second approach called gene suppression. So one can deliver genetic materials like ASOs or mRNAs to suppress target gene. And relevant to Angelman syndrome, we can deliver ASO, right? And ASO clinical trial actually has been advanced to phase 2 and phase 3 soon. So last strategy called genome editing. So in this case, one can deliver genome editing editors into cells to achieve many different ways of gene therapy, like log in, log out, or epigenetic editing. So this technology actually relatively new. It's maybe over 10 years, but actually gained a lot of excitement just over the past few years. Because as Dr. Baudet mentioned this morning, right, for Angelman syndrome, ideally we want to have once per lifetime therapy. So Angelman syndrome potentially could achieve that. Genome editing can potentially achieve one per lifetime therapy for Angelman syndrome. For gene therapy, actually the major challenge has been delivery, how to deliver genetic payloads into the right cells, the right location, right? So in current clinical trials, viral vectors are often used, including many AAVs. So of course, AAVs are good for many purposes. You can transduce cells with high efficiency, could be engineered for cells or organ targeting. And that's why people choose AAV vectors. But for genome editing, AAV probably it may not be the best choice, because then you can imagine, right, you will express a genome editor in cells over decades, and then you don't know what will happen with this bacteria-driven genome editor. They can potentially cause a lot of off-target effect. So as an alternate approach, one can potentially use non-viral approach, non-viral vehicles, mainly including a lot of particles like polymeric particles or lipid particles like RNP. So particularly in lipid particles, again, a lot of attention over the past few years, mainly because of the success of the COVID vaccine. I assume probably all over here probably have received a nanoparticle-based vaccine over the past few years. However, the nanoparticles may not be optimal for deliver genetic materials into the brain. So previous speakers mentioned many times already, right? Our brain is very unique. We have the blood-brain barrier. So this is making deliver whatever payloads through nanoparticles to the brain very challenging, if not impossible. So even for local injection, we can directly inject a payload into the brain, and nanoparticles may not be optimal, mainly because most nanoparticles are relatively large in size, typically about 100 or over 100 nanometer. But in the interstitial space in the brain actually is very limited, just less than 30 nanometer. To overcome this challenges, we develop a chemical approach without use of viral components or nanoparticles or peptides. Instead, we synthesize our chemical molecule STEP. It's about 2 kilodalton chemicals. It's a purely small molecules we attach to each other. So this scheme just show how the STEP technology works. So basically, we can attach STEP molecule to our genetic payloads. It could be protein or whatever. Right now, mainly focus on protein payloads, but we potentially can use a STEP for other genetic payloads as well. So then STEP will bring our payloads into cells very efficiently. So just give you one slide show. This is actually relevant to Angelman program, which Yong-hui is going to talk about in a few minutes. So in this case, we deliver genome editors for disrupting the antisense sequence in the Angelman the chromosome 15. So this genome editor includes 2 components, right? The Cas9 and the guide RNA. And guide RNA guide our Cas9 to the specific location to initiate the cut. So as shown here, we assemble the protein-based machinery in vivo and assemble a Cas with guide RNA. And then we attach our STEP molecule to the surface. In the bottom panel, you will see that in vitro in cell culture, this is very efficient. We can essentially edit most cells. And actually, the machinery, STEP machine, in the engineered machinery is quite stable. We can store the machinery in a freezer for many months without losing activity. I think one unique property or characteristic of this technology is that the resulting machinery, like here, STEP RMPs, are very small, around 10 nanometer in size, negative surface charge, and make them perfect for brain penetration. And Yong-hui is going to show the data regarding the use of STEP RMP in various mouse models. So as mentioned, I'm an engineer on the team. We are not stopped here. Actually, we continue to optimize and develop the system. So in the next 10 minutes, we'll talk about using the current system for deliver to the brain through CSF administration, either intrathecal or RCV or RCM. We're going to figure out the route. So we actually also actively work on developing next generation system for crossing the blood-brain barrier. So we hope we can have the next generation system which can deliver our genetic payloads for Angelman patients through intravenous administration. So actually, Yong-hui is going to show some data. Our current system has the ability to penetrate the brain But I expect we have much we have a system which will be much more efficient for crossing blood-brain barrier. And also the system can be engineered for targeting specific cell types. And also we also engineer step technology for deliver other genetic payloads. So in this case, we will show a lot of data for gene disruption here today. But we also has successfully applied this platform for deliver our epigenetic editors and base editors. In all cases, we found the delivery system is very efficient. And for example, in neuronal cells, they always can achieve over 90, close to 100% of editing. So the next thing Yong-hui is going to talk about the use of this technology for Angelman syndrome application.
Thanks, Jiangbing. I hope from Dr. Zhou's talk you get sort of 3 points. One is the technology is very easy to remember, it's called a STEP. So second, it's very new and very exciting, as Allison put. It's very innovative compared to all other current delivery technology, particularly AAV. By prediction, this could be the safest way to deliver gene editing because we deliver protein. So when this new technology comes down to, oftentimes you can choose which condition to test it. So I relocated from Duke into the Yale. Jiangbing and I actually don't know each other time, but at the time Allison approached me mid of 2020, asking mid of the COVID, asked me to work on the iPSC cell project, generate the new mice. Then I work with the stem cell co-director, and she looked at it, said, okay, you work in the Angelman. She also worked in the Jiangbing. She said, maybe Jiangbing's technology allow you to cure the Angelman. So that's how we get started. So for apply this technology to Angelman. Working last 3 or 4 years, so I truly believe, the team truly believe, luckily I think Angelman clearly is my favorite, I will tell you why. Second, by testing this technology, probably also one of the best model to begin with to test this technology. So I don't need to spend too much time to go over this slide. You heard many different times. Just idea is a reactivated paternal copy gene. As you heard, you can do this antisense oligo, Cas13, small molecule, ATF. Clearly, as Dr. Bodell put on, if it eventually work out, one-time, lifetime delivery enough for cured Angelman, that would be dream come to true. So we take this technology and the first show, as you know, before we go to human, we have many steps to go. First show indeed is working in mice. So let me see the pointer. Oh, okay, there's not a pointer here. No point, that's okay. But just shows you, as you know, and this is a pretty technical slide, and we generated, thank you again for a lot of parents, donated blood, so we generated AS patient-derived cell line. We also made similar to the UB3 YFP fusion mice Dr. Boudin mentioned earlier. We generate the same fusion, CFP fusion to UB3 as a reporter in the iPSC line. So now you can see where you need to just pay attention to the green signal. So after you put a step in the iPSC-derived neuron, almost 24 hours, 48 hours, almost 80 to 90% of the neuron return to the green compared to one below. It's no treatment. And the red signal tells you that's the neuron. So you put together, you say, okay, this is a pretty efficient system in the cell culture system. And the second, and also from the iPSC neuron, initially we call it 2D neuron, 2-dimension. Now we can do called the organelle you heard early. We call 3D neuron. So this is a beautiful organelle. I think it's called cortical organelle. Clearly you can make many different types of organelle. Same thing, pay attention to the green signal, and also shows you dosage effect. If you have low dose, your reactivation is low, and then you have a high dose, reactivation is very impressively high. It's only you dump in and you wash out, and 30 days later and 72 days later, you see the reactivation persistent. And again, all other signal it tells you indeed it's reactivate in the neuron. So all this technology is very, very sophisticated, and luckily Xiaona Lu is the one did all this work. She's here. If you have more technical question, please find her and talk to her anytime during the break. So, so now we move to the in vivo. As you know, we call cell culture models in vitro model. We need to see the in vivo model. So, of course, the mouse model is we begin with. Same thing, you see a lot of similar picture like this, and just highlight the green again. Green is the signal we're looking for. This is done the kind of intrathecal injection first, and then the red signal is highlight is neuron specific. So you can see the day 1, day 2 injection. We also did a day 21, day 42. Of course, the early time is better, but overall, day 1, day 2, you can see the green signal almost entire brain highlight, which means its distribution is very, very efficient. So that's the significant part of this technology compared to AAV. I think AAV is getting better, but for this one, surprising to us, deliver to the mouse brain is so efficient. And we also see that percent of neuron get edited, just one single dose in the cortical cortex region, almost 75% of the neuron became green. And then we know it's a neuron because we stain in the red marker shows as a neuron. We can use other method to sort of confirm this reactivation happen. Remember the earlier question came around like a DNA versus protein. So we indeed shows that the editing happened to the DNA, and DNA, the product, the protein on the right side, which you clearly see is the red column, which is no editing or no incubation, no treatment, it's no protein. When you treat it, the protein increase. Also, as I mentioned, initial testing is an intrathecal injection. We're also working on see if in IV injection, as you know, many of the parents' experience for antisense oligo is clearly quite stressful event overall, but it's a safe procedure. But we're testing whether this technology also good by IV delivery, which clearly will be advantage for a clinical trial. So just very quickly shows you again, IV injection, day 1, day 2, then shows you editing goes to the brain, also pretty efficient based on what we show here. Again, on the multiple different brain region, then you see the green signal is you pay attention to, red signal is the marker to shows in the neuron it's working on reactivated. So clearly we're gonna have to do comparison with IT and ICV because that's a significant implication in the future. Right now we're primarily working on the brain delivery as our project, but in the future we're probably thinking compare these two different methods would be valuable for all the reasons you all know. And clearly, I don't have time to go over a lot of data generated the last couple years, which asking the question, how efficient that single dose delivery can rescue the phenotype in mice, which recapitulate some of the human phenotype. Just highlight a few. You saw the similar diagram from many other talk already. So same as Dr. Siegel was mentioned, just pay attention to what's the compared to wild type, which is low, green line versus the up, it's wild type. So you can see that one dose of a treatment significant rescue multiple different behavior domain. We shows you two here. We also shows the rescue for the learning and the memory and the seizure. We also show deliver different time, day 1, day 2, day 21, day 42. They all pretty efficient overall. Clear that's a difference. Early is better. Just shows you here, Allison shows you this video before we provided, but this is a little bit different. Stress that one is on the right. The left is deliver day 1, day 2, postnatal day 1, day 2 mice. On the right side, it's a deliver 42 days old mice, which is equivalent, 42 days old mice equivalent to the human adult individual. So therefore, we think this technology indeed at different time point, as I know some of the parents, some of your kids are young, some are older. So overall, we see the benefit, at least in the mice. We have to move on to the larger animal, and also most important in the clinical trial in the future, that we'll do that. So clearly, we continue working many different, but this is a project we're starting as you learn, we got a large support from NIH last year. We started this project earlier this year. It's a larger team effort. I will not be able to go individual and tell you what each party is doing for this project. Just shows you on the left side, it's a lot of a company we engage in to produce this new product. A lot of challenges, but this company are also excited to help this project. Clearly, there's many other components, the CIO, Charles River, and the Yale, FAST, and the NIH for sure, Rush, and Courageous, all working together. In some sense, we team working, almost meeting regularly, weekly, and Jennifer Allison from FAST, typically at least one meeting every week. Sometimes Jennifer and Allison, we meet 3 or 4 times per week. But it's a huge team effort together. But we are very excited, making good progress since the project began almost 11 months ago. Clear, also, I cannot go on to highlight detail for all the progress we made for each highlight here. That will take about a couple hours. So Alice only gave me 20 minutes, so therefore I would just highlight this a few things here. We have very successful interact meeting, and NIH program officer, we will say, oh, this is a textbook interact meeting we run. Thank you again for Jennifer for this great effort. Her expertise make a huge difference for this success. And we work very closely with a lot of vendor and company to produce this very special material. A lot of challenge, but we are making a progress. And we are design and conduct an IND enabling study that will take next at least 18 months to maybe 2 years to finish. And we also develop or design a very unique and innovative study, use the PET imaging to look in the biodistribution. That's in progress. And we pretty much complete for the research dosage range of funding study, then in the mice, and we are moving on to do this one in the good quality of material in the rodent and the non-human primate. NHP is a basically monkey. We also pay attention to, for sure, in order to move from the human safely, it's very, very essential for any CRISPR editing technology, make sure we correct the Angelman, but we now create another extremely rare genetic condition. But so far, all the assessment, we've done quite extensive assessment with people with great expertise working together. So far, we have not seen any significant concern for consider we call off-target event. So they're all good, and just in conclusion, clearly this is a very exciting project we are undertaking. And there are many, many challenges for sure. We hope this STEP, again, it's the STEP delivery platform. It's innovative, and if we can work it out, they will be potentially revolutionary, not just for Angelman, for many, many other neurogenetic conditions. Because it's novel, complex, we anticipate we're going to meet a lot of challenge, but the team are working very, very hard. So, and also working together, that's the key. I think the spirit Allison mentioned multiple times for this community, working together is the key. Individually, you can do something, but for the project like this, it has to be the multiple party and the team working together. And now we hope to reach the goal for that finally in the next couple years. So finally, I want to acknowledge the people I want to acknowledge. First, I want to acknowledge Dr. Baudet, and as you know, I was a student in his lab almost 20 years ago. Thank you for him to introduce me to the science for sure, because I came from China and I know little for the science, and also introduced the Angelman project, which is, he did not share with you. I did not start working with Angelman. I started working on the GABA beta 3. I made the first knockout very successfully for GABA beta 3, but we realized someone did it 6 months earlier than me. So then we said, okay, let's choose the E6AP. So that's the history. Thank you for all the support and the continued support. I hope next couple of years we're going to deliver your vision, one dose, lifetime, sufficient enough, cure the Angelman syndrome. I will not go to all the other acknowledgement. Clearly, a lot of work is done in Jiangbing's lab and my lab. And as I said, Shauna is here, and the data I presented, many is generated by her, and then you can talk to her anytime during the break for particularly interesting, more technical detailed question. It's a larger team. I will not go over one by one, but to highlight Conrad from the Courageous, Allison and Jennifer. That's a big team. Thank you again.
So we've learned a lot about drug development today, and what I can tell you in the last few years for me, that program is incredibly complicated, but it is so exciting. And seeing, you have a gene therapy where you have one construct you're making, or an ASO, you have one construct you're making. This is making 3 constructs to put them together for one final construct, and you have to understand how each of those 3 things behave independently to then understand how they behave together. In drug development, that is very, very complicated, but it's looking amazing, and we're getting there, and we're moving as quickly as we can. So we hope to deliver upon our Arch dream as well. But in the meantime, there's a lot of other exciting things that are likely going to be hitting the clinic and hitting humans first, which is also exciting. We'll hear a lot more about that tomorrow.